Thank you for coming this afternoon. I'm really glad to see you here today. I welcome to the third lecture in our Whom You Love series. I don't want to take a lot of time to talk about the project overall. I'll just remind you, you can find out everything you want to know about this project by going to whomyoulove.com. And thanks to the marvelous Casey McGovern, I can't believe it. Stumbled on that. We have a great website that you can take a look at and find out about future speakers, etc. Also, I want to point out to you that in the back we have some resources from the LBGT Resource Center, and there are also these lovely cards that give you a preview of the rest of the symposium series. I'm especially happy to introduce Dennis McFadden today because for several reasons. First of all, I can tell you a little bit about Dennis. He started making his world in California, and would eventually, after getting his bachelor's degree there, went to Indiana to go to get a Ph.D. in psychology. And it wasn't long after that before he was the head of a position at the University of Texas in Austin, where he's been for the vast majority of his career. He has been extraordinarily productive. He has a 15-page CV. I threatened that I was going to read all the titles to you, but I better not do that. And he's also been very successful in getting grant support from NIH over these years, including among some of the reasons I'm jealous of him is that he got a Jacob Javits Award from NIH, which is a very difficult thing to do. It's only given to the most outstanding researchers. And the important thing he did for me in terms of this series is it was Dennis's work that first made me think that there might be a biological influence on human sexual orientation. Up until about 1998, I was quite skeptical that there was such a thing. And so I'm especially happy to have him here and talk to you about that. So if you would please welcome Dennis McFadden.
The top little button. Thank you, folks. I appreciate the introduction. I appreciate being introduced by Dr. Breedlove. He's one of my longtime science heroes. We are going to talk some about the physiological evidence about the origins of sexual orientation today, but the fact of the matter is it's going to take a while for us to get to that part of the talk. I'm going to spend a considerable amount of time at the front end making some general comments about a number of different things, some terminological things. I want to make sure that everyone understands what the origins of the two sexes are. We're going to talk about a couple of natural experiments that have been provided with us, maladies of exposure to hormones during prenatal development. These are CAH and AIS, and we'll talk about those pretty soon. I also want to spend a little time talking about what you require as evidence for the innate origins of sexual orientation. Then we'll finally get into the auditory system. We'll do a little background, then we'll talk about the work that I've done. Some of the goals for today are to try to convince you that there is, in fact, a considerable amount of evidence that non-heterosexuality does have a biological basis, is innate. And along the way, I hope to convince some of the experimentalists in the audience that some of the measures that we're going to talk about today, otoacoustic emissions and auditory evoked potentials, have the potential to be valuable supplemental non-invasive tools for people working on a whole array of different topics pertaining to human development and sexual differentiation. So, take a step back and we think about the world, and we note that there are some people who prefer as sex partners people of the same sex. And there are some other people that prefer as sex partners people of the other sex. Why is that? Did these people just make a decision at some point in life and then live their lives in accord with the decision that they made? Or might there have been some biological, some genetic, physiological contribution to sexual orientation? And if there was some kind of physiological, biological contribution, can we get some insight as to what, in fact, those mechanisms were? Before I get too deeply into the talk, I want to make clear to the audience that I know that talking about straights and gays or heterosexuals and non-heterosexuals is a great simplification of the situation. Yes, there are people who have live lifelong, strictly heterosexual lives, and at the other end of the spectrum, there are people who live lifelong homosexual lives. But in between, there's a whole array of folks. People who live a homosexual life for a time period and then live as a heterosexual for a time. There are people that have a bisexual orientation for an entire lifetime. There are people who are asexual. There are people who cross-dress. There's a wide array of folks between those two extremes, I admit right at the front that using any kind of dichotomy like heterosexual and non-heterosexual is a simplification, but in order to just have this talk, we've got to be able to have some terminology, and so that's the distinction I'll be making. So an important assumption that we all need to understand before we get too deeply into the topic is that there is this central tenet of modern neuroscience that whenever you see a difference in behavior, whether you're talking about differences between two groups or talking about changes in an individual across time, if there are differences, there must be some corresponding difference in the structure or functioning of the brain somewhere. How could it be any other way? If I teach you some new fact about the Civil War, and tomorrow and 3 weeks from now and 6 months from now, you can reproduce this fact for me, a fact that you did not know before, pretty clearly something is different inside your brain to allow you to have access to that fact, to integrate it with other facts you've got, etc. Similarly, if it's a motor response, if you learn to have a better backhand in tennis, and 6 weeks from now and a year from now, you're able to implement those changes in your stroke so it's smoother or whatever. Clearly, something has to be different in the brain from what it was before. That's pretty obvious, but we need to get it said up front. We know that there are a number of structures in the brain that differ between the two sexes. Here are some examples. Actually, I don't want to linger on this slide very long because there's some really, really tasteless stuff lurking in here. As we talk about group differences over the course of the talk, whether we're talking about sex differences or differences between people of differing sexual orientations, we're going to remember that we're talking about differences between the means. We're not talking about individual people, we're talking about group effects. Here's some imaginary data on some kind of a test where we've given the test to both males and females. You can see that it's possible for some males to score 80 on this test. It's just that it's more likely that females will score 80 on this test. Similarly, it's possible for some females to score 20, but it's much more likely that the males will score 20. There are differences in the distributions. We'll be talking about differences in the mean, but knowing the score for some one particular person in 80, you can't with great confidence know whether that person comes from group A or group B. I'd like to suggest that if we're going to look for evidence of possible biological origins of non-heterosexuality, that a logical place to begin, it's logical for me at least, is to talk about the mechanisms that produce the two sexes in the first place. One of the defining characteristics of non-heterosexuality is an atypical choice of sex partners. So before we get into the discussion of the data on non-heterosexuals, it makes sense that we make sure we understand how we get the two sexes in the first place. This is kind of a review of, I don't know, junior high school or high school biology for most of you, but just indulge me. For all females, every cell in the body has two X chromosomes. That includes the germ cells. So when the germ cells split and form two eggs, each one of those eggs has one X chromosome in it. If all the cells in a male body have an X chromosome and a Y chromosome. So when the germ cells split and produce sperm, half of the sperm cells are carrying an X chromosome, half of them are carrying a Y chromosome. Because of that, there's 4 possible combinations that can occur here. Obviously, any combination that leads to 2 X chromosomes in the fertilized egg means that the person is destined to be female. If everything works out according to the plan, that person will end up female. Similarly, if the fertilized egg ends up with an X chromosome and a Y chromosome, then if all things work out according to the program, you're going to have a male. Oh, this advances. Yeah, cool. Now, if you're talking about an XY fertilized egg, an egg that's headed off to be a male, what happens is that cell divides, then there's 2 cells, and then those 2 divide, and there's 4, those 4 divide, there's 8, etc. And eventually you build up an entire body. Somewhere pretty early in that process, in the male developing embryo, a gene gets expressed. This gene resides on the Y chromosome. It's called SRY. And when that gene gets expressed, what happens is a collection of cells in this developing embryo, cells that are eventually going to be testicles. Now they're just the testes. They're just a collection of cells inside this developing body. Those cells are activated by virtue of this SRY gene being expressed, and those embryological testes begin to produce androgens, testosterone, the male sex hormone, so-called. The hormone circulates around in the body of the fetus, and it masculinizes various target organs. So in humans, as in all males, in humans as in all mammals, the developing male fetus masculinizes itself. The SRY gene turns on the testes begin producing these androgens, and that's what leads to the eventual differences in body, brain, and behavior in the males as opposed to females. If there's no SRY gene, and there is not one in any fertilized egg that has 2 X chromosomes, because the SRY gene is resident on the Y chromosome. If there is no SRY gene, there's no androgen production, and the embryo develops as a female. This is the origin, part of the origin of the cliche that in humans, females, as in all mammals, female is the default condition. A complex series of events have to occur to produce a male, otherwise you get a female. It's not quite as simple as that, but for these purposes, that's fine. Now, some additional facts to know about this whole process is that the surge of androgens that occurs during the middle part of gestation for the developing male comes to an end before birth. As we'll see in the following slide, after week about 24 of development, the androgen levels fall down to a baseline. They're basically identical to what's present in a developing fetus, of the female fetus of that same age. At birth, the hormone levels in males and females are identical. Any differences that you see in males and females at birth are attributable to things that happened before that, not things that are going on right now. For a few months then, after birth in a male, there is another surge of androgens that lasts for 5 or 6 months. There's apparently some additional masculinization that's going on of the body, brain, and behavior. And then the androgen levels fall back down to baseline, and then they remain there until puberty starts. So any differences you see in small children between the 2 sexes are not attributable to differences in the circulating levels of sex hormones at that time. They're all after effects attributable to things that happened during earlier development, much of it during prenatal development. A fact that we need to get out on the table straight away is that adult heterosexuals and adult non-heterosexuals don't differ in their circulating levels of sex hormones. They are identical. So if we're going to look for any kind of hormonal contribution to sexual orientation, we've got to look at a different, some different place in development. Early on, prenatal development is a logical place to look. And this just then, this complex slide, all you need to look at is down here. Here are the data for testosterone. I don't know if you can see from the back, but from about weeks 8 through 24, there's this first surge of testosterone in the male fetus. There's birth right there. And before you get to birth, you see the androgen levels are at a baseline level. There's a so-called second surge that lasts for a few months. And then these levels are at the baseline, just exactly the same as they are in females. And then comes puberty and all hell breaks loose. One of the important concomitants of what we're talking about is if female is the default condition, then male is the default choice for sex partner. The typical female prefers males for sex partners. Typical males prefer the opposite sex, females, as the non-default sex as for sex partners. So the implication is that something happened during development to change the male preference from the default choice to the opposite choice. Something had to be different in the brain. Something had to get masculinized, to use a word, in order to have the typical male choose females as a sex partner, because that's not the default choice in humans. So, while we don't know exactly what that change is, we don't know exactly where in the brain it is, it's pretty clear, given this central tenet of neuroscience, that there's got to be some kind of a change that occurred probably during prenatal development. Note in this context that homosexual females exhibit the male typical choice of sex partner. If there's some kind of switch in the brain that gets flipped from the default state to the non-default state in males, it looks like that also happens in homosexual females. It looks like the switch inadvertently got thrown. Somehow they're behaving as if something got masculinized in the brain. For homosexual males, they exhibit the female typical choice of sex partner. It's as if the switch didn't get thrown. It's as if some collection of cells somewhere in the brain, we don't know where, didn't get masculinized, the switch wasn't thrown. It's a way, in this context, to think about non-heterosexual behavior. I want to talk about a couple of different errors of development that occur. One syndrome that's of interest to us is called androgen insensitivity syndrome. In this condition, you have an XY fertilized egg, and the embryo begins to develop toward being a typical female at the right place in prenatal development. The SRY gene gets expressed, the testes start to get active, androgens start to be released into this, this developing embryo, but they don't have functional androgen receptors. Those various collections of cells around the fetus don't take up the androgens because they just don't have the androgen receptors. So the whole process proceeds down the female typical route. Female is the default condition. At birth, you have someone who looks perfectly female. Later in life, these people are attracted to males and marry males, have perfectly happy lives living as males, even though as females, even though they are XY. So the message this communicates to us is that this exposure to the androgens is truly a critical stage of development of both the body and the brain. And it's one of the other major facts that contributes to the idea that in humans, female is the default condition. The other condition I wanted to talk about is congenital adrenal hyperplasia. This is a situation in which the adrenal gland goes bad during prenatal development, and as a consequence, the developing fetus gets exposed to very high levels of androgens. If it's a male, nothing much really happens to the body. There's some downregulation of some sort, and the developing fetus kind of protects itself against this. They still have to be treated after they're born. They've got some metabolic problems that have to be treated. But the really interesting case is if it's a female who has CAH and gets exposed to high levels of androgens, because at birth, her genitalia are intermediate to those of typical males and typical females. She's characterized as being intersex. She has an elaborated clitoris. Many of these infants are operated on soon after birth, and the genitalia are return to a more female-looking appearance. Internal sex organs are reasonably normal, and many CAH females later do marry and have children. But in addition to the masculinization of the genitalia, they're different in a whole pot full of ways. They, from a very early stage in life, they prefer rough and tumble play, like males, unlike many females. They're more interested in boys' toys very early in life. Give them a choice between a truck and a doll, they'll take the truck. There's an array of different behaviors that you can see there that are masculinized. And the message we get is that these non-genetic effects, these effects of the androgens operating on this developing fetus, changed a whole bunch of things in this person's body and behavior. The importance is androgens during prenatal development. So, if we're going to go looking for evidence of biological origins of non-heterosexuality, we have to ask the question of what counts as evidence for such. Here's a list of differences that have been reported in the literature between heterosexuals and non-heterosexuals. There are some brain structures that differ in size between the two groups. The strength of the hands, the ability to accurately throw projectiles, the ability to mentally rotate three-dimensional objects in space, the ability to navigate through complex environments. These are all things that are different in non-heterosexuals and heterosexuals. Typically, these measures, like, say, throwing accurately projectiles, there'll be a sex difference. Males will be really good at this task, females less good. Navigation, males better than females. And then the homosexual and bisexual females are intermediate. They're in between on these measures. And the question is, which of these findings is convincing to you that there are innate differences between heterosexuals and non-heterosexuals, differences that exist at birth due to physiology? I'd like to argue that none of them is acceptable. The reason is each one of these could be an acquired characteristic. It could be, from what we know about modern neuroscience and changes in the brain, it could be that the changes that exist in the brain structure are themselves results, not causes. A person in some way goes off and lives a different lifestyle, a non-heterosexual lifestyle, gets different kinds of experiences from what he would have gotten as a heterosexual. And what's going to happen? The brain's going to be different. Similarly, with these other measures, the ability to rotate objects, the ability to throw projectiles, if a child went off in a non-traditional, atypical, not sex-typical direction, he might get exposed to a number of different things or not get exposed to certain kinds of things that makes him better or worse on some of these different tasks. accurate projectile direction takes some practice. And if a kid is disinterested in ball games and whatever, might not get the experience, all of these things could very easily be acquired differences as opposed to innate differences. But I'd like you to consider these. It's the case that homosexual males and females are both more likely to be left-handed than are heterosexuals. It's called non-consistently right-handed just to kind of capture the whole range of the differences, but basically left-handed. Homosexual males begin puberty about 3 months before heterosexual males. Homosexual males have larger penises by about 1/3 of an inch than heterosexual males. One of the topics you're going to hear about in a few weeks from Ray Blanchard, you don't want to miss that talk, is about the fraternal birth order effect, so-called. The fact that for every additional for every additional older brother a male has, the chance of his being homosexual goes up by about 1/3. Now you start with a kind of a small number, 2% to 5%, somewhere in there. So 1/3 more than 2% only goes to 3%. You know, it still stays a small number, but the chance of being homosexual goes up with each additional older brother that your mother gave birth to. To me, that really does feel like an innate difference. Something is going on there in the mother, presumably, that's leading her to provide a different intrauterine environment for the later conceived males. These differences on this slide, sorry about that, are for me much more convincing evidence of an innate contribution to non-heterosexuality than were the ones on that first slide. Well, how in the world can experimenters or real people distinguish between innate and acquired differences? I'd argue that it isn't easy. One way is to just use common good sense. Does it seem likely that some particular characteristic is acquired rather than innate? That's basically the nature of my reaction to the list on the previous slide. It just seems very, very, very unlikely that something about a person's lifestyle could alter when their puberty started, or the size of their penis, or the fraternal birth order effect. These things look to me like pretty good evidence for some kind of innate biological contribution. Another strategy, a really good one experimentally, is to study infants and young children. Look for differences on various kinds of measures in those children and wait until they grow up and see if which ones become heterosexual, which ones become non-heterosexual, then go back and look at the data that you had on the various measures that you took on the children and see what predicted those things. Unfortunately, prospective research of this sort is almost never done because it's god-awful expensive. The rate of non-heterosexuality is very low, so you have to run really, really large samples, test really large samples in order to get any kind of reasonable fraction that are going to be non-heterosexual. And it's just an enormous amount of work. It's certainly no way for a young academic to get promoted. But the point of raising it now is to make clear that you understand that there is something of a difficult problem of distinguishing what constitutes an acquired and what constitutes an innate characteristic or difference. So finally, we're getting to the auditory system. There she is, isn't she beautiful? This is what I've spent my life studying. This, of course, is a frontal view. You have a buzzsaw that goes through the head, and the front part of the head falls away, and it leaves behind this view. There is an external ear canal that's terminated here by the eardrum. There's an air-filled space behind that. The first bone in the 3 bones of the middle ear system is attached to the backside of the eardrum. That means then as the eardrum moves in and out in response to air pressure changes that exist because there's a vibrating body out there in the body, in the world, you have these air pressure changes. The drum moves, the bones move, and that means the final bone in the series, this 3rd bone, moves in and out of what is a fluid-filled space back here, the spiral cochlea. And this is what eventually leads to hearing. This is now a closer up view of what we just saw, and we've moved the plane of the buzzsaw back just a little bit more so that when it cut through, it sliced off part of the spiral cochlea. And you can't see in this rendition very well, but we see that we would see, if you could see it better, the canal is made up of 3 separate chambers, 3 separate distinct regions. And so this next slide shows the cross-section there at one of those cuts that were revealed. We end up with 3 different chambers because there are 2 membranes that divide up the system. This is a membrane that you know of from high school or before. This is the so-called basilar membrane. This is the membrane that gets put into vibration when sounds occur out there in the real world, and that third bone of the middle ear system goes in and out of the fluid-filled space. This membrane goes up and down in response to that, and it eventually leads to our rich experience of hearing that we have. Sitting on top of the basilar membrane is the organ of Corti, and in the organ of Corti are some receptor cells, the cells that turn vibratory activity into neural activity, the coin of the realm for the brain. There's 2 different types of hair cells in there, 2 populations. This population out here in green is the so-called outer hair cells. We'll use that term later. There's 3 rows of those that mark all the way down the length of the spiral cochlea. Something on the order of 12 to 16,000 of them total. And then there's another single row of inner hair cells, and they run along in parallel with them. There's only about, say, 4,000 or so of the inner hair cells. The outer hair cells have our attention for today because they have this very interesting property. As the basilar membrane goes up and down, and these little hairs that stick up off the top, the cilia off the top, get sheared back and forth, what happens is the outer hair cells are so-called electromotile. They change their shape. They get longer and shorter in response to the shearing on the stereocilia. Because they're held tightly in place at the top and the bottom, this change in their shape changes the local micromechanics of the cochlea. The short story is these cells, the outer hair cells, act like amplifiers. The magnitude of displacement of the basilar membrane up and down is greater because of these outer hair cells. They actually amplify the mechanical displacements that occur. And because of that, we are able to hear the weak sounds that we can hear in the quiet. Your hearing sensitivity, your sensitivity to the weakest sounds is attributable to the effects of these outer hair cells amplifying the displacement of the basilar membrane. I hope an interesting fact for some of you is that the auditory system doesn't just process incoming sounds and turn them into the experience of hearing that we have, but the normal, intact cochlea makes sounds. These are called otoacoustic emissions. They're very weak. In order to record them, we have to have a relatively expensive small microphone that we stick into the ear canal with a foam tube around it to get a really good seal. And it's with that system we're able to record these really weak sounds coming out of the ear. We're going to talk about 2 different types of otoacoustic emissions today. There are spontaneous otoacoustic emissions and click-evoked otoacoustic emissions. First, spontaneous. These are basically pure tones that are coming out of the ear all the time. They're different in frequency in different ears, and there's different numbers of them in different ears. The way we measure them is we put that microphone in the ear canal and we record like 2 minutes worth of sound from the ear canal. We then go through and do some processing. We get rid of the noisier moments. It's a noisy place. There's a heartbeat, there's breathing, the jaw moves around. The external ear canal is a very noisy place. We get rid of the noisiest time periods and then analyze the rest of it, and we get what is known as a spectral representation. The spectral representation shows the strength of the sound present at a lot of different frequencies. This ear does not have any spontaneous otoacoustic emissions. It's just representative of like the background level that we record in the ear canal. This ear does have one really sharp, large, spontaneous otoacoustic emission, somewhere in the vicinity of 1,700 cycles or so. And then this ear, as you can see, is flooded with otoacoustic emissions. If we have any luck at all, we have an audio demonstration of this. First, we'll hear the ear that has no spontaneous otoacoustic emissions. Nothing much to hear there, you're just getting that for reference purposes. That's what it sounds like in the ear canal, except we've amplified it quite a bit. Yeah, I think you might have been able to hear a few episodes of some breathing or some jaw movement in there. And compare that then with this. I think some of you can hear that there is a tone superimposed on that background noise. And if you didn't hear that one, then you are surely going to hear this one. Oh, this is the sound recorded from the ear canal of a young woman. She's flooded with spontaneous otoacoustic emissions. The second type of otoacoustic emission we're going to talk about today is the so-called click-evoked otoacoustic emission. This is like an echo. We put in a very brief sound, a click. It goes through the middle ear system into the cochlea, and then after a few milliseconds, a waveform comes back out. It's like an echo coming back. And these waveforms are different in different ears. They have different configurations, different durations, etc. Here are the click-evoked otoacoustic emissions of the same ears we just heard for spontaneous. There's 5 presentations, the click and then the response, the click and then the response. Didn't hear much of a response. That was just mostly the click. Here's the other one. I think you can hear that has a little bit of tonality to it. And then here's this. So though we don't really listen to these sounds, we process them, analyze them. What we do is we measure how big these amplitudes of displacement are around the middle line and characterize the click-evoked otoacoustic emissions that way. Here are some characteristics of otoacoustic emissions. They're more prevalent in females than they are in males. Something on the order of 75 to 85% of all females have at least one spontaneous otoacoustic emission. Males, it's more in the vicinity of 50%. In terms of click evokes, they're stronger in females than they are in males. Overlaid on this difference between the two sexes is an ear difference. Otoacoustic emissions are stronger and more prevalent in the right ear than they are in the left. The reason I bring up those various facts is the differences between the sexes, the differences between the ears that exist in young adults exist in newborns. Everything we know about otoacoustic emissions is that they are very constant through life. They can change, but they're most of the time they're very constant through life. The implication of that is the otoacoustic emissions that I measure in a young adult are going to be very representative of what I would have measured in that ear at the time of birth. Another fact to emphasize, there's 2 important facts to emphasize here. One is that hearing sensitivity co-varies with otoacoustic emissions. So people that have lots of otoacoustic emissions also have good hearing sensitivity. This is just hearing pure tones in the quiet. The other thing to note here is there is just nothing about either spontaneous otoacoustic emissions or click-evoked otoacoustic emissions that any person could change by making some kind of choice in life about how they were going to live. Here's an illustration of the sex differences. Here is spontaneous otoacoustic emissions on this side. You see the difference between the 2 ears, right being stronger than left. These are the females. These are the males, again, right being stronger than left. Those of you who know something about effect size from experimental psychology, the effect size is about 0.98 for the spontaneous otoacoustic emissions. Here are the same data, big sex difference and the ear difference in the click-evoked otoacoustic emissions. Again, to remind you that we're talking about group differences, means. Individuals are spread all over, both sexes are spread across the spectrum. We don't know very much about we don't know all there is to know yet about the mechanisms that produce otoacoustic emissions, but what we do know is that these outer hair cells we talked about a little while ago, these cochlear amplifier cells, these electromotile cells, are critically important to the production of otoacoustic emissions. If you lose your outer hair cells for one reason or another, and you will if you continue to listen to music at those sound pressure levels, you are killing outer hair cells. And the way you know you're killing outer hair cells is that they scream at you. You can hear them dying. So stop doing it. You don't regenerate them. Once you kill them, they're gone. And that's really difficult for those of us who do experiments, because when you kill off your outer hair cells, you take away our otoacoustic emissions. And you make our lives harder. So there's multiple reasons to be careful with your hearing. The important part here is that otoacoustic emissions are dependent upon these outer hair cells for their production. So this is a little boxology that to help you organize some of the comments made so far. What I'd like you to think about is that both otoacoustic emissions and hearing sensitivity are both results of a common cause. That's having these cochlear amplifiers. The existence of the cochlear amplifiers is crucial to both of those other two measures. Well, why is there a sex difference in the otoacoustic emissions of newborns? Why do males have weaker otoacoustic emissions at the time they're born from females? Well, I don't actually know, but I would argue that a really good first assumption would be that it's exactly the same mechanism that's affecting all the other differences between the sexes in body, brain, and behavior. It's the exposure to androgens during prenatal development that's at the heart of it. Specifically, what I'm suggesting to you is that high exposure to high levels of prenatal androgens works to diminish, to weaken the cochlear amplifiers, and as a consequence of that, you have weakened otoacoustic emissions and reduced hearing sensitivity. What got me into the business of sexual orientation and the auditory system was a study we did on twins. Some of you know that experimental psychologists study twins as a way of getting some information about whether a trait is highly heritable, attributable to genes, or not. And the nature of the reasoning is that if there's a strong genetic component, then when you measure pairs of identical twins, they ought to be more similar than pairs of non-identical twins. And the reason is the identical twins have exactly the same genome, all the same genes, same chromosomes, same genes. So they ought to be more similar than the others. Then you get those data and you do a little arithmetic and you get a measure called heritability. That's what I got interested in. What's the source of these individual differences in otoacoustic emissions? Why do some people have so many and others have so few, and they're so weak and some people stronger than others? Might it be something to do with genetic, might it be heritability? And so we did this experiment, and we got nice measures of heritability. For those who care, it's about 0.75. It's a very healthy value. But what caught my eye more than the heritability measurements is this fact. In the course of this study, we not only ran monozygotics, which are identical twins, and dizygotics, which are fraternal twins, we also ran some opposite-sex twins, male-female pairs. And what you see is that the otoacoustic emissions of females with male co-twins are like those of other males. What we know about otoacoustic emissions is that they're very constant through life. We know that sex differences exist at the time of birth. The strong implication here is that these differences in the otoacoustic emissions of opposite-sex female twins existed at birth. And the question then becomes, how come? Are there any other ways that females with male co-twins are masculinized? Yes. And here's a list of them. Don't have time to talk about them in any detail. Well, these are made available. Dr. Breedlove will make them available to you later. So, I would like to suggest that this difference between females with male co-twins and females with female co-twins might very well be attributable to exactly the same mechanism we've been talking about so far that's produced other differences between the sexes, etc. Androgen exposure. Might it be the case that for some reason females with male co-twins get exposed to higher than normal androgen levels during prenatal development? If so, then we would have the result that we see. But how might that happen? Where would those androgens come from? The answer is from the developing male co-twin with which she's sharing the intrauterine environment. This sounds kind of fanciful. This sounds kind of off the wall. Why should the androgens in that developing male affect this female? In fact, there's an enormous literature on this in other mammals, non-human mammals. It's called the intrauterine position phenomenon, and it happens. Females get during the intrauterine environment, females get masculinized by virtue of the exposure they get to the males also developing in that womb. And there's no reason to think that it couldn't happen to human females as well. And you saw from that previous slide that there are a number of ways in which opposite-sex female twins are masculinized in addition to our otoacoustic emission thing. So it's the same argument that we've made before. I'm trying to argue that the opposite-sex female twins got exposed to high levels of androgens. It weakened their cochlear amplifiers, and they had weak otoacoustic emissions as a consequence of it. Finally, we're getting to the meat of the talk. After working with the opposite-sex twins and thinking about androgen exposure, etc., I began wondering about what other kinds of special populations we might study, and non-heterosexuals came to mind, and we did a study. We measured the otoacoustic emissions in both heterosexuals and non-heterosexuals, and we got data like these. These are the data for the spontaneous otoacoustic emissions. At the far left here, we have heterosexual females, the difference between the left and the right ears. You've already seen that, you know that fact. Over here on the far right are the heterosexual males. So there's a big sex difference between heterosexual males and females. But that's not new. You already knew that. What's new is this. Homosexual and bisexual females have otoacoustic emissions that are shifted in the direction of heterosexual males. Simply as a descriptive term, the otoacoustic emissions of homosexual and bisexual females have been masculinized. Those are the spontaneous otoacoustic emissions. Here are the click-evoked otoacoustic emissions. Same basic story. Here are the female heterosexuals. Here are the male heterosexuals. Big sex difference. You already knew that. Here are the homosexual and bisexual females. They're again shifted in the direction of the males. Purely as a descriptive term, their auditory systems seem to have been masculinized in some way. When? I don't know, but I think a reasonable assumption is sometime during prenatal development. Another type of measurement that we've made is called auditory evoked potentials. These are brain waves. You put electrodes on the scalp, a couple of electrodes on each earlobe, and then a couple of other electrodes on the scalp. Then you present sounds like clicks to the ear, and you measure the brain waves that result. They look something like this. This is what happens before the click is presented, just a bunch of random wiggles. The click is presented right here at the upright, and then over the course of the next 10 milliseconds or so, there's a series of great big waves that occur. We can measure the latency, the time from the click to the peak, or we can measure the amplitude of the peak of these brain waves. And we've done that. What's interesting about auditory evoked potentials, like otoacoustic emissions, is that the sex differences that exist in these measures exist in newborns as well as in adults. So the differences in their brains existed at birth. And also AAPs are reasonably consistent across time. And there's no obvious way that someone could, by choice, change her auditory evoked potentials. What are you going to do? Hold your mouth like that? It's not going to work. So here, we had 19 different measures, latency and amplitude measures. 5 of them showed differences for the homosexual and bisexual females. And what we see is effects that are very similar to what we saw before. In this particular measure, it's an amplitude. The heterosexual females have a weaker amplitude than the heterosexual males. There is a sex difference. And then here are the homosexual and bisexual females. You can see they're shifted in the direction of males. Once again, we see a masculinization effect. One more wave. This also is an amplitude. For this particular one, the amplitude is larger in the heterosexual females than it is in the heterosexual males. There's a nice sex difference. Once again, the homosexual and bisexual females are shifted in the male direction. They've been masculinized. It's not just the cochlea, it's the auditory brain that's getting changed. And for the first time in anything I've shown you, there's a difference for the homosexual males. There weren't any in the otoacoustic emissions. For the first time, there's a difference here. What you see is that the homosexual males actually have a weaker, a smaller amplitude for this particular wave than the heterosexual males. It's as if they've been hyper-masculinized. They're even more male-like than the heterosexual males on this particular measure. Had I a little more time, I'd talk to you a little bit about lifestyle differences. Might there be something that's going on in the lifestyles of heterosexual and non-heterosexual people that lead to these differences? I don't think so. If somebody wants to ask me a question about that later, I'm happy to talk some about it. What this slide reveals is that because of my belief that neither otoacoustic emissions nor auditory evoked potentials can be altered by some kind of choice made by their owners, I would like to argue that they represent strong evidence for some biological contributions to sexual orientation. What we don't know yet is exactly how those androgens affect the cochlear amplifiers, but the strong implication is that they do. Here are some of those differences that we saw at the beginning about physiological differences that I think are revealing about the innate origins of homosexuality, things that it's very hard for me to understand how they could be changed by choice of their owners. One of them I didn't mention is called atopy. This is a propensity to have various kinds of allergic reactions, rhinitis, asthma, hives, hay fever, much more common in non-heterosexuals than in heterosexuals. We talked some about the other ones. I've added one down here that wasn't there before. This is a behavioral one, not a physiological one, but it pertains to what goes on in early life, early childhood. There are things that go on, particularly in the lives of people who later become gay males, that are different from what happens in the lives of children that become heterosexual males. Their toy choices, their preferences for clothes, etc. There is some predictability from this behavioral measure rather than a physiological one. I wanted to tell you about localized effects. I don't have time. I'm very hopeful that somebody will ask me a question about that later. One of the fantasies I've had for 20-odd years now is that we'd be able to get a collection of congenital adrenal hyperplasia females to measure their otoacoustic emissions. And I've been frustrated 2 or 3 times at this, but it is an ongoing study, and the initial data are very encouraging. The otoacoustic emissions of those women are masculinized just the way the theory, the explanation I've been offering predicts they should be. I wanted to end today with 2 slides that talk about some historical landmarks. I'm going to be 72 in a couple of weeks, and during the course of my life, there have been some really remarkable changes in society. This documents some of them. I suspect that many of the youngsters in the audience don't have an appreciation for how recent a lot of these things are. baseball not being desegregated until the late 40s. Americans for Disability Acts is relatively recent. Some of these other things, Civil Rights Act, come from the 60s, but it's relatively recent that these events occurred, social changes that prevent you from discriminating against people for some particular reason. This parallel list is milestones for reproductive and sexual life. There are two yellow ones. Those are bad things that happened. Alan Turing is like the father of the modern computer. He was a mathematician in Britain. He was caught in some homosexual activities, forced to take estrogen for a year or more, and the changes in his body, brain, and behavior were more than he could take, and he killed himself. That's a bad one. Here's one from 1986. Sodomy laws were upheld. That's pretty darn recent for the US Supreme Court to be upholding those laws. The rest of them are basically good things that have happened. Homosexuality stopped being a psychiatric disorder only in 1973. Prior to that time, it was a psychiatric disorder by definition. Somewhere on here. Oh, contraception. It wasn't until 1965 that women were allowed access to contraception by law. Some states allowed it, but a lot of states prohibited it. Massachusetts didn't let you have contraception until 1965. Down at the very bottom of this list, I have one that's undated because it's yet to happen, and that's the issue of same-sex marriage being legalized. Also shown for each of those is how it happened, whether the Supreme Court did it in a decision or whether it occurred in some other way. My wife says that if I were honest, this would be the title that I would use for this talk. And this is what retirement looks like. So I'm sorry I went a little long, but Dr. Breedlove did talk too long at the very front end, claiming numerous pages of a CD that he made up. Tradition in Texas is to let those go who need to go, have appointments elsewhere, and we'll wait 10, 15 seconds for that, and then we can open it up and start answering any questions that you might have.
I know there are going to be lots of questions, and let me ask you, please don't be bringing the microphone so everyone can hear.
Thank you, and I do have a fun. 3 quick things. Thank you. I'm older. Thank you for showing these young people those laws, and let's pray they're not taken away from us. Young people, get out there and vote. They might be, but thank you for that. Secondly, very quickly, I have 5 brothers, and the oldest one might be the gay one, and all the other ones aren't. And so that just breaks, you might want to study my brothers now. But, and the third thing is I appreciate, I'm not in a class here, I'm just an older public, lady coming in from the public who has a gay son.
Welcome.
If I get it, thank you, and thank you for this. If I get any information from you, I really appreciate, from the professor. But thank you for explaining that, because I've had many people say they think it's all environmental, so thank you.
Well, now you've got grounds to counter-argue.
I think you might have missed a yellow one on your 2 lists ago. Don't Ask, No Child 1993
I do it wrong.
was not, which I think should have been yellow as a bad thing.
Oh, well, I'm sorry. I accept that. There are people that, I'm sorry. That's that's a legitimate point that on the slide about the sexual orientation right, right, that this probably ought to be yellow too, because there are a lot of people that take offense at that. Then why not just be open about it and later it became so hate crimes. Yeah, it's been repealed since. So absolutely, you're right.
So I noticed that on some of the slides with the graphs that there were some interesting characteristics about the columns for bisexual males. In particular, differences, a much larger difference between the left and right ear for the spontaneous otoacoustic, I forgot what the E stands for, emission, and that similarly in the brain wave, the wave 5 amplitude, that there was a bigger difference in bisexual males between the left and right than in other categories. Is there anything known about why that might be? It seems really interesting.
No, I'm sorry I didn't mention that in passing. I was feeling kind of rushed, but the answer is I don't have a lot of faith in those data. If you'll notice that the ends were much smaller than they were for the other categories, I'd want to have a lot more data before I put any weight into that one. The other thing to say about this is that there's a substantial number of gay males and experts on homosexuality, which I don't think of myself as, that are kind of disbelieving about the category of bisexual for males. They think that this is a situation that's a temporary situation, that they're still not, they haven't faced up to the inevitability of being homosexual and like that. It seems to be more of a phenomenon of younger people than for older people. I don't have a real position on this. I just think the audience ought to know that there is absolutely no concern about the existence of bisexuality for females. There certainly are bisexual females, but there's some question about whether males that claim to be bisexual really are.
And we'll have other folks.
And then there'll be people talking about it. Yes. Keep coming.
Okay, my question is, since you said that having twins that are fraternal, one male, one female, causes the female to have male-like...
Otoacoustic emissions.
Yeah, right. So is there, or shouldn't there be, like, a very large statistic of females who are bisexual and homosexual to have male twins?
Yeah, and the answer is they don't seem to. There is some evidence of more homosexual fantasies than in, singletons, but that's not the right comparison. The right comparison is the same-sex dizygotics. So female, but fraternal. That's the best comparison because they're both twins. They both had a partner in the interuterine environment, and so that's the better comparison. So there is some evidence of it, but it's not strong, and I think the implication is that it pertains to the degree of exposure that they got. For some reason, this is the most bizarre part of this. For me, I grew up studying the auditory system. Why in the world should the auditory system be so sensitive to these apparently small differences during the prenatal environment and the differences in androgen exposure? I mean, this is just truly bizarre to me. But you point out a nice one. I mean, you would think that the brain would get more masculinized in those people than it is. There is some evidence that the dentition changes. They're not as they don't have as many children. There are a number of things that are masculinized about opposite-sex female twins, but the brain is not strongly affected when it comes to sexual orientation. And why is that? I don't know, but my suspicion is it's the degree of exposure, how much. It could also be timing, note. It could also be the timing of the exposure.
So your data suggests that homosexuality in women it may be related to prenatal exposure to testosterone, but as far as homosexuality in men, you showed the data that suggests, if anything, you're hypermasculinized. So could you talk a little bit about that apparent, paradox, or how you think about that?
Thank you. It's curious that when you look across a wide array of different measures that have been made on homosexual males, you'll find some measures on which they are hyper-masculinized. They have a larger penis. They have earlier puberty. There are a number of ways in which they're hyper-masculinized. Then there is a bunch of measures on which they're not affected at all. And then there's other measures in which they're hypomasculinized. They're shifted in the female direction. How in the world can this be true if simple androgen exposure is the whole story? Everything ought to shift in the same direction if degree of androgen exposure was the whole story. I talked about degree of androgen exposure mostly as a simplification, just so we could get through the talk, but in fact, I don't really believe in it. My suspicion is that the androgen levels are not so terribly different. My suspicion is that there's differences in the uptake of the androgens that are available at different places in the brain, what I call localized effects. You could implement this by having differing numbers of androgen receptors on certain collections of cells at particular times during the development. That would do it. That would explain it. This problem, it doesn't exist only for the human sexual orientation data. There's an enormous literature in which people have studied the effects of hormones on non-human mammals. And you can go read these papers. They'll give injections of extra androgens to young male newborns, and then measure a whole bunch of things and wait a while, and then go measure a bunch of things. And some of them are hypermasculinized, just like you'd expect. You'd expect if you gave an animal extra androgens, it would be more masculine than it would have been had it just been exposed to its normal level. And then there are some things that aren't any different at all. Some sexual behaviors that are just exactly, it's as if you didn't give that injection. And then there are some in which these animals are hypomasculinized. It exists not just in the male gay data, it exists in the animal literature. It doesn't get talked about very much in the animal literature. I think what happens is the experimenters see something where it just doesn't fit with the way they're thinking about it. They gave extra androgens, you should see hypermasculinization, they talk about that. And the fact that one or two measures were hypomasculinized, well, it's just kind of error of measurement, that kind of thing happens, it's a chance event, and they don't talk about it. But I've looked across a number of different studies and I find, so gerbils have a very characteristic so-called tripodal stance. They stand like this and they put one hand down. And the females put that hand down, the males put that hand down. Give males extra amounts of androgens, they put that hand down. So they've been exposed to higher levels of androgens, but they show the female typical response. They've been hypomasculinized. So I'm sorry, more answer than you wanted, but.
Is it okay if I ask 2 questions really quick, or no?
Well, it's $5 more for the second.
Oh, okay. Is there any evidence of females with older brothers being more?
No, absolutely, it doesn't work that way. And you'll want to, when you come to hear Ray Blanchard's talk in a few weeks, he'll talk about that for sure. No, it's as if what happens is that the mother's body has some memory for the previous male pregnancies. Think about it. A developing male in a female body is something of an insult. This is something that you've got to protect yourself against if you are female. And what do females do? They produce continuously high levels of estrogens all throughout the pregnancy. And I think that part of that is to defend themselves against the insult of androgens being introduced into their body by the developing male fetuses. And Dr. Breedlove is more of an expert than I on this, but the point is that no, the fraternal birth order effect doesn't work in females, presumably because they don't stimulate this immune reaction that apparently is being stimulated by the succession of male pregnancies.
Okay, and for fraternal twins, the male, where you have a male and a female, why do the females, even in a single pregnancies, have androgen receptors?
Sure. Both sexes need some of each in order to mature normally. What in fact a uterus produces is testosterone. It just gets immediately aromatized into estrogen. So there are some low levels of androgens.
Okay, it's just, it would be more that when they're male, female fraternal twins, that the female is exposed to more androgen because of the male fetus, like hypothetically.
That's my proposal to you, yes. And if you don't like my proposal, that's perfectly fine. Come up with another explanation. Because it's not just the otoacoustic emissions that are masculinized in OSDZ females, it's those other things on the list. You've got to explain all those.
And we know that females have androgen receptors before birth because that's how they respond to CAH when that happens. So we know this.
why they get masculinized.
My question was about the other things on the list. You have a slide that starts with dentition.
Yeah.
Could we see that again?
Well, only if you're not going to ask a hard question.
And you've got a copy. Oh, okay. And these other topics will come up in future lectures. There was someone else who was down here. I think we've got time for two more.
Okay, thank you. We saw a slide that showed the patterns of levels of androgens prenatally and up until puberty. And then you mentioned that there's a wealth of research on opposite sex dizygotic animals females particularly, who have increased levels of androgen prenatally. And so I'm wondering if there's any research that shows that humans, opposite sex dizygotic females, have increased levels of androgen in the womb. If there's any
Right. No, I don't know. I don't think anybody. No, I don't know how you would do that. I mean, you'd have to go in and do the what do you call it? To get the fluids, amniocentesis, to do that, to get the fluids, and then you have to keep them, and then you have to analyze them and everything. So I suspect it'll be a long time before we get that answer. No, it's a speculation. I'm sorry about that, but it's a speculation. But it's not so far to seek because it's fully believable that the same processes of diffusion that operate for the non-human mammals, where the androgens diffuse out of the body into the intrauterine fluids, that ought to be operative for the human developing male too. You know, there must be data on that. Somebody must know if the amnio fluids of just say a male, developing male, that's got to be, that fluid's got to be higher in androgens than a developing.
It is.
Okay, well,
And for individuals with CAH, the androgen levels from amniocentesis are higher than for same-sex individuals without CAH, for example.
So you should have had an expert computer.
In your future research area, you had something about autism?
Yeah.
Is there any?
I had a fantasy, no, just didn't work out. I tried to collaborate with a number of people, and the granting agencies didn't like us. I think there was a goodly amount of turf warfare. The people that are controlling the money for autism didn't like the idea of somebody coming in with a new measure, somebody, just a psychologist that has no history and whatever. This is me whining. But the answer is we tried. We had a number of grant proposals, and they failed. The reason for being interested in them is that it's primarily a male effect. The vast majority of autistic kids are male. It smells like some kind of an anomaly during prenatal development. 2, many of the symptoms of autism are expressed quite early in life. So again, it looks like they are after effects of something that occurred during prenatal development. That's why I was interested in autism. And
Also, are they auditory?
Well, I mean, let's see. So, and there's this idea that autistic individuals are hypermasculinized, and so the simple question is, do you see evidence of hypermasculinization in the otoacoustic emissions in autistic males?
Right.
And the answer is,
we'd love to make that measurement, and you're.
no one wanted to pay to
The NIH didn't think it was a worthy question to fund, so we won't know the answer unless somebody else picks up the... I've got equipment that you can borrow if there's a youngster here looking for a career.
Without funding. Sometimes there's a penalty to be paid for asking.
We did study ADHD kids. ADHD males, one type, so-called inattentives, are hyper-masculinized on their otoacoustic emissions and their finger length ratios.
I wanted to hear about the slide you didn't have time for.
That was the localized effects.
That was
I
the
did that one,
localized effects.
but I do appreciate it.
Any other? All right. Well, I'm sure there's going to be more questions afterwards. So let's thank Dr. McFadden.
And I will hang around for a few minutes if you want to come down and talk about things.